And the final point in interpretation is something called as a disease mutation map. Okay. This is something that is actually pretty nice mechanism to interpret what is happening and utilize the power of the DeepBind model. Okay. So, the DeepBind model basically says that given any S, there is this black box that will compute this f of s and give you this prediction f of s, right. If you now make certain mutations to this S, and so you get this S hat, which is the mutated S, then you can use the same DeepBind model. So, this is the DeepBind model, right, DeepBind model, this f, and you can get f of s hat. Okay. If f of s hat is very similar to f of s, then you know that the mutation has not done anything to the binding properties of the sequence. But if f of s hat is very different from f of s, right, if these are very different, then you know that the mutation that is introduced, into S to convert it into S hat has a deleterious effect. It either increases the binding strength or decreases the binding strength. Okay. Using this kind of point mutations and then running the exact same model on these different mutated sequences can reveal a lot about what is the effect of certain disease causing mutations on the binding strength of these motifs. Okay. So, that is what are called mutation maps. So, just to make it a little bit more clearer, you have the original sequence S, okay, you have the mutated sequence S hat where some letters are changed, okay, specifically only one position is changed to a different base. Okay. If you have an example here in the next slide, so if you have GAATA, then maybe the second letter is changed to a C. Okay. So, this is changed to a C, everything else remains the same. So, this is your S, this is your S hat. Okay. Then your importance score of this mutation, the effect of this mutation is calculated using this concept of delta Sij, okay, where you are looking at the probability of the sequence S hat to bind the protein minus the probability of the sequence S to bind this protein, right, in terms of a classification problem. And this probability of S or probability of S hat, we have seen before that this is delta, this is sigmoid function of f of s, right. So, f of s is going to be some value between negative infinity and positive infinity, and this p of s is going to be just this sigma of f, right, and your f of s is going to be between this minus infinity and infinity, and this is 1, this is 0. Okay. So, this p of s is the probability of the sequence to bind the particular protein of interest, okay, using the model for that protein. And then you have this term maximum of 0 comma p of s comma p of s hat. This is just to make sure that if none of these two proteins bind, then we are not interested in this delta score. Okay. If at least one of the protein binds at a reasonable probability to the target protein, then you give it more weight, that is this max going to do, and then you are going to look at the delta of the probabilities of the sequence S binding to the protein minus the sequence S hat binding to the protein. Okay. And essentially, so then in this particular case, if you change this, this is S and S hat. So, you are saying that if P of S hat is, let us say, some value 0.9, okay, and then P of S is going to be some value 0.1, okay. S was not binding to the protein, now S hat with the C change is now binding to the protein. So, there is an increase in binding strength, right. So, your delta S value is going to be pretty high, okay, because the delta S value, if you remember, is P of S hat minus P of S. So, in this case 0.9 minus 0.1, right, into max of 0.9 minus 0.1 into max of 0 comma 0.9 comma 0.1, okay. So, this delta S value right. So, for this particular case it is going to be this. So, that is going to be 0.8 into 0.9, okay, that is going to be 0.72. So, 0.72 is going to be this value of changing GA to GC here, okay, and when GA had 0.1 and there is an increase in probability of 0.9, okay. So, positive scores means you have an increase in binding affinity when you make the mutation, negative scores means you drop in the binding affinity. Okay. So, this kind of analysis can now be applied to real protein sequences, right, real transcription factors and then real gene sequences. Okay. So, there is a gene called LDLR. Okay. In this particular case you have a gene called LDLR, right, and there is a particular motif upstream of the gene in the promoter region of the gene, and then there is this transcription factor called SP1 that binds to this. Okay. In a wild type, right, you have this motif, it likes binding to this region in the promoter where there is a lot of C's with some interspersed T's. Okay. Let us look at this particular location alone, this really huge C. So, this C is really tall, so it kind of gives the indication that that is an important location. So, now if you mutate the C into anything else, either an A or a G or a T, right, you get a blue score, right. Blue means negative values. Okay. So, you can see here that this is the delta S scores, positive values are shown in red, negative values are shown in blue. Okay. So, these blue means there is a decrease in binding strength. Okay. So, if you go from the C to any other letter indicated as a D here, A, G or a T, then you decrease in binding. Okay. And then so you basically SP1 never binds again. So, this dislodges off if the C here gets converted into a D. Okay. So, that is why it is called as SP1 loss in LDLR promoter. So, this gives you very fine grained prediction of what the effect of mutations are. Okay. So, there is specifically a mutant where the C changed to a G in a particular disease, a metabolic disease that is related to cholesterol metabolism, okay, where the C in this particular location changes to a G, and what this is saying is that when the C has mutated to a G as in this particular disease, we have lost the interaction or regulation of LDLR by SP1. Okay. This is just one example. There is also an example in cancer where you look at a gene called MYC. This is a very famous cancer related gene, and then you look at a transcription factor called TCFL2. Again you have this MYC gene, and then you have some enhancer that controls. Enhancer is just simply a substring that is nearby that, a motif that controls this, where if the transcription factor comes, this TCF7L2 comes and binds, then it will either activate or repress the, it will regulate the production of this MYC mRNA, right. And then so you can see here again that we can look at this particular location G here, right. This in terms of the height of the logo diagram, this is not as tall as the C that we looked here, right. And if the G is mutated into anything else, right. So, you have this A here, C here, G here and T here, right. If the G is mutated into A, C or G, it is mostly white here, but if it is mutated to T, it is little bit blue, okay, not as dark blue as in the other case. So, if this mutation G is mutated into a T, then there is a loss of transcription factor binding, and so the control of this TCF7L2 transcription factor on MYC is weakened, okay, and that is what this showed. So, going from here to here, you have these blue colors here, okay, and then if the T changes back to any of these other things like C and so on, then you see that red color here. This is just the opposite effect, okay. Again, the point I am trying to make is that there are many such mutation maps for disease causing mutations on effect on these different motifs, and this gives you a very fine grained look at what are the exact letters that are along the genome, the exact positions in the genome, exact motifs that are controlling nearby genes, and these are genes involved in diseases. Okay. So, you have this fine grained view of exact locations within the motifs and those motifs within the sequences where these disease causing mutations can disrupt transcription factor binding and disrupt the regulation of transcription factors on the downstream genes. Okay. And it can also point out the cases where you can introduce mutations to get back that function. Okay. So, for instance, in this mutation C to G, then it will increase the binding. So, for a mutant G, if it converts back to C, then you will increase binding. So, this gives you both the problems with disease causing mutations and ways to fix those problems using specific mutations on the enhancer or promoter sequences of target genes. Okay. So, to conclude, just citing from the paper, DeepBind uses deep learning to analyze how protein binds to not only DNA such as in transcription factors, but also RNA, right, in terms of RNA binding proteins, and allowing it to detect mutations at this fine grained level and positions, right, position motifs and positions within those motifs or these mutations that could disrupt cellular processes and cause disease. Okay. So, this is kind of a nice summary of what is in this paper and what we have seen in this session. And again I am just ending with some of the points I mentioned earlier on why we were looking at this particular DeepBind model. It is a representative starting point of machine learning and specifically a deep learning solution to the long standing motive finding problem, which is an important problem in computational biology, due to which we have seen it multiple times. And I hope at the end of the session you see value in taking a deep learning approach to this whole problem. And there are many more works that have come off after this DeepBind work or around the time of this DeepBind work, and some of the recent works look at how to interpret these models, right, more than mutation maps, trying to look at what the parameters of the model mean and what implications it has on the geometry of the DNA sequences where the transcription factors bind to. Okay. So, not only understanding the effect of disease causing mutations, but you can also look at what is the information about the geometry of the helical structure, for instance, of the DNA that are in the motif binding regions versus those that are not near the motif binding regions. Okay. So, the geometry of the DNA near the binding sites can also be informed by these deep learning models. So, the field is very vast for you to explore. So, if you find it interesting, you should definitely take a look at those papers. So, thanks for your interest and time, and we will conclude the session here. Thank you.